good afternoon, and I appreciate being here with you today. These are not my slides, so if you would just back up one. I didn't bring slides because when I first spoke with June about talking to patients today, we discussed having slides, and I thought it would be nice just to have a conversation to tell you a bit about Fulcrum and a bit about the molecule that we're developing, and not to present slides on the data to you today. Fulcrum is a company of around 50 scientists. We've been in existence for roughly 2 years, but 2 years prior to that, we did very serious and very deep thinking about the company and about what we were going to do. We intended to hire a group of scientists with a lot of experience in drug development, and in fact, I have more than 20 years of experience, as does the majority of our management team. Our focus was to be on monogenic diseases, or diseases that were caused by the upregulation of a gene inappropriately, such as DUX4 and FSHD, or the absence of the expression of a gene, such as something called Fragile X protein in FMRP in a neurologic disease, Fragile X. Our concept is that we would go to the root causes of the disease to modulate the expression of that overexpressed protein. We would work to bring it down, or to upregulate the protein that was not expressed. We would work to bring it up. We have experts in transcriptional regulation, and you heard a lot about transcriptional regulation today. You heard that genes are turned on inappropriately for a lot of reasons, and the DUX4 protein, as you've heard, is the protein that causes the pathology in FSHD, and its inappropriate expression in muscle is driving the disease. So as we formed Fulcrum Therapeutics, and you wonder why we might call ourselves Fulcrum, well, if you think about a fulcrum, it's about manipulating a weight over a fulcrum. And our concept was we were going to try to bring down the high level of DUX4 expression and balance its expression in the muscles of patients. How would we seek to do that? We wanted to do it with an oral medicine that can be taken and that can systemically reach through the entire body and to every muscle that would be affected. It would, of course, need to be safe, and it would need to be effective. And we became very focused in patient interests, and we acquired tissues and cell lines from patients. We established novel methods to grow them in culture in our laboratories so that we could actually generate muscle from FSHD patients that were fused into myotubes and that were able to constrict in our tissue culture settings. So we established some beautiful models for screening. We undertook the screening of small molecule chemical moieties to see if we could find those that could reduce DUX4. We also took genetic approaches, and you heard a bit about CRISPR-Cas9 earlier, and we used that as a genetic tool for manipulating proteins that we thought could be important in DUX4 regulation. What we found were a series of molecules that could modulate DUX4. And we went on and we did a lot of evaluation of those molecules, and we came down to a clinical candidate. What I didn't tell you is what my role in the organization is I'm the head of the translational group. We have a young scientist who's head of the early development of molecules. We have a neurologist who's heading our clinical studies, and I'm sitting in between those, translating the findings from our early discovery team and the cell lines that we study into the neurologic understanding of how we would bring these molecules into the clinic. We work very closely as a team. The company invested in early translational development because we realized that if we didn't understand this disease exceptionally well, we would not know how to test a molecule in a clinical setting. So what we did is we undertook relationships with experts in the field. We went to Rabi, we went to Jeff Statland, we went to other researchers, and we said, we want to work with you, we want to use your expertise, we want to learn from you, and we want to contribute. And we've spent the first 2 years while we've been developing our small molecule, also developing our concepts of how we would test these molecules clinically. So we've come up with a molecule, it's called FTXA at this moment. We're still in preclinical stage, and we're planning for clinical evaluation of that molecule in FSHD patients next year. This year, what we're intending to do is a feasibility study. That feasibility study is going to tell us a lot, and it intends to make us successful next year in our clinical evaluation. We're going to be recruiting patients to small studies at a number of sites in the United States and in Europe. We'll be doing a number of biopsies, and I encourage you to think deeply about biopsies, because when we think about reducing DUX4 protein in muscle, the only way that we can ask if we've done that is if we actually go in and biopsy the muscle ahead of our therapy and then after our therapy. We intend to use needle biopsies, and they're very well established in this field, and we're working with the experts who have done it historically. We're looking for characteristics of DUX4. What you didn't hear from this group is that DUX4 itself regulates gene expression. And we can't detect DUX4 very well in human muscle in FSHD patients. And so what we're intending to study is not DUX4 itself, but its effects on other gene transcripts. We're working with experts in the field to say, what are those gene transcripts? How can we best measure them? Which are most often expressed in FSHD patient muscles? I also want to talk a little bit about the diagnostics question that was addressed to Rabi, to Dr. Twill earlier on. We need patients to have a formal genetic diagnosis in order to be able to enter them into a clinical study. So if you don't have that formal diagnosis and you wish to be part of a clinical trial, I encourage you to get that. It'll be required before you enter. For Fulcrum, if patients come and don't have that, we will pay for that and we will help them to have that in order to be entered in. So we have a molecule that reduces DUX4 transcript and it reduces DUX4 protein. It also reduces the amount of cell death that is seen in FSHD muscles in culture when we carry muscle myotubes in our culture dishes. And we are going to be evaluating it in a limited number of animal models and then working towards going into the clinic. I want to say lastly is that we're very integrated into the community. We're working closely with the Clinical Trials Research Network scientists, and we're encouraging them to take on additional studies. So you've heard a lot about the 6-minute walk test, but we also know that there's a lot of upper body issues with FSHD. And we've gone to that group, as Dr. Statlin mentioned to you, and we've asked them to engage and consider upper body measurements as well, which are range of motion measurements, some without weights and some with weights, in order to put stress on the muscles in order to be able to really reveal the physical effects of FSHD. And with that, I will thank you for your attention.
Thank you. Our next speaker is Monica Hayhurst Bennett from Genea Biocells.
As an earlier speaker said, this is not kind for short people. I'm going to stand to the side. So I want to tell you a little bit about Genea Biocells. Genea Biocells is part of the Genea group. They're actually a fertility clinic, and it's one of the leading IVF clinics, and it's been around since 1985. So what Genea Biocells has been able to do is amass the largest commercially available bank of normal and affected human embryonic stem cells. And we've done that because in our fertility clinic, patients come that carry genetic disorders, and they do not want to sometimes they don't want to pass those on to their children. So they come, they have IVF done, they donate embryos that they know have their genetic mutation, and then they are able to transfer and have healthy children with their genetically normal embryos. So we've gotten disease-affected embryos that way, and as well patients that have grown their family and have remaining embryos sitting in our bank and no longer want to transfer them have donated them as well. So we have a very large bank of both diseased and healthy embryos that we can work with. So Genea Biocells is originally located in Sydney, and in 2015 we moved to San Diego. And we really changed the focus of the company when we moved there. And we changed our identity more to a therapeutic development company, and we're really focused on neuromuscular diseases. And one of the valuable things that we've done is develop these stem cell-based platforms. And in particular, we have a platform where we can take a human pluripotent stem cell, so that embryonic stem cell is that kind of cell, and in a dish we can differentiate or convert that cell into a mature muscle cell that is multinucleated and can even contract. So we use that platform to build disease in a dish models. I see. Okay. So in 2011, because we were based in Australia originally, we had a good relationship with FSHD Global. They decided that it was really important for the research community to develop a stem cell-based model of FSHD. So they funded us to generate embryonic stem cell lines from FSHD patients. And so that work resulted in 3 FSHD1 embryonic patient stem cell lines. So we used those FSHD1 patient cell lines. We applied our platform and we converted them into muscle cells, and then we studied them as mature muscle cells. And that actually resulted in a paper in 2016 where we described both the lines and our platform. And we were very happy to see in our disease in a dish model, many of the pathologies that we see in FSHD patients, we could actually just see them in our disease in a dish model. And in particular, like the analogy from this morning, we saw the Joker arise, so we see DUX4 expression. We see all the bad things that the Joker does as well. So it turns on these downstream targets of DUX4. We see cell death in our disease in a dish model. And so that we then moved on and decided this was a good place that we could screen small molecules. So we performed a screen where we specifically looked for molecules that could turn down DUX4, and that led us to what we call GBC0905, and I'm just going to tell you a little bit about what we know that molecule does, at least in our in vitro setting or in our culture dishes. So both in our model system as well as tissue that we have used from primary biopsies, so this is patients' biopsies, both FSHD1 and FSHD2 biopsies we got access to, we see that our compound actually turns off DUX4. And then it turns off all the downstream targets of DUX4. And that's really important because we believe that turning off DUX4 is going to be curative in FSHD. It also rescues the muscle cells from cell death. And then it does that in combination without having any sort of toxicity in our cultures, and as well as really not affecting the muscles. So what we get when we treat our muscle cells is we get healthy muscle that doesn't express DUX4 anymore. So, I've put in here where we think our compound is playing a role, and it's really just turning off DUX4. So how we think that our molecule will probably work in patients is it will stop the progression of the disease. So I want to tell you a little bit about the status of this drug right now. We've recently just received orphan drug designation, and this is a designation given by the FDA that recognizes new therapeutics that are for rare diseases, and it's going to assist us as we move on to clinical trials. And right now, our timeline is that we plan on having interactions with the FDA and applying for a new investigational new drug application. And our target is to actually have this drug in the clinic by 2019, so basically the end of next year. And that's it. And so I think the most important thing I want to say is I really want to thank the patients, because all of this work has been made possible at Genea from patients. We started off with these donated embryos from FSHD patients. We've been able to receive materials from academic repositories, and so we're endlessly grateful for the material that you've helped us or helped provide so that we could do this work. We're also extremely grateful to the foundations in the FSHD community who've catalyzed this work at Genea, and as well as every patient and every clinician and academic who has talked to us and really helped to mold our program.
you.
Thank you.
Our next speaker is France Sverdrup, and he will be talking about another small molecule screen for DUX4 toxicity, or to suppress DUX4 toxicity.
Thank you. I'm from St. Louis University, and I'm working with a drug company called Ultragenyx Pharmaceuticals to develop a drug to suppress DUX4 expression. And I want to tell you a little bit about focus on BET inhibitors and how we're using a mouse xenograft model for drug discovery. So BET inhibitors block DUX4 expression. BET stands for bromodomain and extraterminal, or BET family of proteins. There's 4 genes in this family, BRD2, 3, 4, and T. And the genes code for proteins. These BET proteins are known for activating gene transcription or turning on genes. And this is shown over on the left where you have the promoter that Scott talked about. And BET proteins, in this case BRD4, is shown in red there, binding to acetyl groups on the chromatin and promoting the gene transcription, turning that gene on. BET inhibitors actually bind to those little pockets of the BET proteins and prevent the BET proteins from binding to the promoter and therefore turning off the gene. It's known that BET proteins bind to D4, Z4 repeats and the DUX4 gene. I should emphasize here that we're talking about hundreds of proteins that bind in this region, and it's been very difficult to discern what all the proteins are and how they turn on DUX4. But BET inhibitors block DUX4 expression in differentiating FSHD muscle cells. And here's a little schematic showing that with an increased drug concentration, we turn off the gene. So with Ultragenyx, we are using medicinal chemistry to optimize novel BET inhibitors to suppress DUX4 in FSHD muscle cells. So we use a xenograft model of FSHD to ensure that we're using human epigenetic regulation of the DUX4 by introducing human cells into the mouse. Human FSHD myoblasts are grown in culture, injected into regenerating mouse muscle, and those FSHD myoblasts contribute to regenerating myofibers. And you can see that outlined in red is human-specific spectrin staining, indicating that those myofibers contain the human cells that are engrafted. So the important thing is that we maintain the human FSHD genetic arrangement and DUX4 epigenetic regulation, because that's what we're trying to suppress. DUX4 and DUX4 target genes are expressed as the muscles differentiate in vivo. That's shown here over on the right side, where after injection of the cells begin to differentiate and they upregulate the DUX4 gene, as well as a target gene, MBD3L2. So we use this as a pharmacology model so that we can treat these animals systemically with drugs. The drug needs to get to the muscle and turn off DUX4. So this model is really enabling us to do quite a bit. So the first thing is pharmacokinetics. We want to make sure when we treat the mice with the drug that the drug not only gets into the system, but it goes into the muscle, and it's there at a high enough concentration for a long enough period of time to have the effect on DUX4 that we hope it will have, which is suppressing DUX4 expression. So shown here is treatment with a tool BET inhibitor where we can suppress the 2 DUX4 targets, MBD3L2 and LUDX, with drug treatment with the mice. Interestingly, we found that when we treat the mice for 14 days with BET inhibitors, that we have an increased number of the FSHD cells there at the end of that treatment period. And so this is an interesting finding that we're actively following up on. And safety is also important. We want to make sure that our muscles develop normally while we're treating with BET inhibitors. MyH2 is a marker, myosin heavy chain of late differentiation. And also, we can show that the BET inhibitors do not inhibit the generation of myofibers containing human spectrin. So this summarizes what I said. I just want to go down to the bottom. We're evaluating both repurposing candidates, which will be much quicker, as well as novel compounds for therapeutic development. We're comparing multiple mechanisms in this mouse model to compare, because we only want to bring the best compound forward that's safe and effective. And I've had large number of collaborators, and I want to thank the FSH Society for funding in the past, and also Ultragenyx Pharmaceuticals. Thank you.
Okay, we're running behind, so I'm going to hurry up and be quick here. Our next speaker is Charis Himeda, and she'll be talking about CRISPR-based repression of DUX4.
Okay, I'm going to tell you very briefly about our work using CRISPR technology as a potential therapeutic approach for FSHD. So CRISPR is a gene editing system that's gotten a lot of attention in recent years due to the power and the relative ease of using it to modify the genomes of organisms. And most of the controversy surrounding this system has come from the idea of using it to modify the human genome, especially in ways that can be passed down to our offspring. This is not a new idea or a new concern, but CRISPR has made it much easier to do than ever before. So I think it's important to point out that what we're doing here is not this. We are not modifying eggs or sperm or embryos in any way. We are simply trying to use this system to correct muscle cells in FSHD. So the normal system uses a guide RNA, which binds to the Cas9 protein and recruits it to a specific location in the genome. So you can design your guide RNA to target, for example, the mutated dystrophin gene in Duchenne muscular dystrophy. And Cas9 will then cut the dystrophin gene. And if you provide a correct copy of the DNA, the cell's normal repair mechanisms will insert the new copy in place of the old. And this cutting system is what the vast majority of researchers are using to try to correct diseases. But this is not an ideal system for FSHD because, as you know, the disease locus is just one single repeat unit with hundreds of nearly identical repeats throughout the genome. And cutting your genome in hundreds of places is probably not a great idea. So an alternative system is CRISPR inhibition, and here the Cas9 protein is mutated, so it's incapable of cutting, and it's fused to a repressor protein. So now the guide RNA recruits a system that represses DNA instead of cutting it. And this is really ideally suited to FSHD, since we want to turn off or dial down DUX4 expression. And importantly, other similar repeats in the genome are normally repressed, so recruiting a repressor to these other locations shouldn't pose any unnecessary risk. And if the repression is stable, it can be inherited by subsequent generations of cells, but importantly without making any permanent changes to the genome. So in 2016, we published a proof of principle study showing that CRISPR inhibition can reduce pathogenic gene expression in FSHD. We used muscle cells from an FSH patient and showed that CRISPR inhibition could return the FSH locus to a more normal repressed state and significantly reduce expression of DUX4 in its target genes. And the important point from this study is that CRISPR targeting of the locus is conceptually possible, which was not a straightforward question. And we're currently testing CRISPR inhibition in our lab's mouse model of FSHD. Okay, so what are the challenges for CRISPR technology as a therapeutic avenue? There are some general challenges which apply to all diseases. Can we deliver the therapy to all of the target cells? Will all of the target cells be modified if we do? What happens if this system is recruited to other unintended places in the genome? This gets at the idea of off-target effects, which are understandably a serious concern. Can CRISPR components stimulate an immune response in patients, which would be a problem? And then, of course, can the system actually provide a long-term cure? And there are some FSHD-specific issues, which really are things we still need to learn about the disease. These are how many muscle fibers actually need to be corrected. Do muscle stem cells need to be corrected? And how much of a reduction in DUX4 expression will actually provide a therapeutic benefit? So as a therapeutic strategy, CRISPR is still in its infancy, but this platform is constantly evolving and being improved on because there are a lot of people working very hard on it, and it's potentially very powerful and very exciting. I just want to thank our lab's funding sources, and this is the Peter Jones Lab at the UNR School of Medicine. Thanks.
Okay, the next two talks will be discussing using methods to target the DUX4 messenger RNA. I talked about that in my earlier presentation. First speaker on that is Yi-Wen Chen, and she'll be talking about antisense therapy for DUX4 targeting.
Thank you. And thank you for giving me this opportunity to talk about antisense oligonucleotide strategy used for treating FSHD. So this method has been used for have been a lot of different trials and studies trying to use this to treat neuromuscular diseases. And then recently there's one approved, FDA approved for Duchenne and then SMA. Okay, so this actually is FSHD cells in this picture. And you can see there's a single cell and there's a nucleus in the cell. And right beside it, you can actually see a myotube. That's actually a long structure with multiple nuclei in there. So, so this is actually a cell culture, the cells cultured in dish. So as earlier speakers, Dr. Belleville and also Dr. Harper mentioned that the DNA that keeps the information of our genetic information actually is in the nuclei or nucleus here in the single cell, and the nuclei in that string, the myotube. And messenger RNA actually is made from the nucleus, the DNA in the nucleus, and then ship out into the cytoplasm out of the nucleus. And as you can see that there's a genetic code in there. There's the A, G, and C, and U in this case because this is an RNA, and the A will pair with U, and the G will pair with the C. And this is actually an example of the RNA that would be like for the DUX4. One thing I want to point out is you can see the backbone under all the letters, and this is actually how they actually link all these codes together. So that's when people usually talk about backbone. And for the RNA, it's actually, that's because in the backbone, there's the sugar, it's a ribose sugar. And so that's why it's called RNA, ribonucleic acid, and there's a phosphate group at the bottom. And actually, that's how the RNA is linked together. And then the DNA is deoxy. So you can see, if you can see the differences between these two, there's one missing oxygen. Well, there's one's OH, for the RNA, and then the other one is H in that figure. So it's a remove an oxygen. That's the reason it's DNA, deoxy. Okay. So this is the messenger RNA, and the antisense therapy basically utilize or take advantage of natural mechanisms in our cells. So we didn't really invent anything. It's actually what our cells are doing. We just take advantage of that. So what happens is when there is a single strand RNA in your cell, and for example the DUX4 transcript, and then if you have a DNA strand that's complementary to it, in this case you can see the first letter A, and then you have T at top, and then basically they complementary with each other, and they actually can bind, they can hybridize, they can bind tightly with each other. And in that case, for example, for therapeutic purpose, we can put a small stretch of this DNA molecule, and then if we can deliver them into the cell successfully, then they can actually search for the sequence that's actually complementary to it. In this case, they actually can achieve a very specific target of the RNA they want to reduce or knock down. So they, in a way, one of the advantage of antisense therapy is actually to have a more specific knockdown. So you only knock down DUX4, not something else by accident. And when that happens, there's an enzyme inside your cell, and here RNase H actually will recognize this DNA and RNA duplex, and then they will degrade the RNA that's associated with your antisense molecule. So that's basically how it works. There's a lot of practical problems. For example, if it's a natural form, it's not very stable, and it will be degraded quickly. So there are additional modifications that's chemically to the natural form of RNA. So there's one example, that's what I'm going to talk about later. It's LNA. So you can see there are two different arrows, red arrows there, actually tell you how it's modified. And you can see one of oxygen is replaced with the sulfur, and then the other one is actually a different linking pattern. And then basically these improve the stability of this molecule, and it actually can stay in the cells longer for therapeutic purpose. So you don't need to have it every hours or something like that. So for the study, we actually use a mouse model. This is where we are at the moment, from Peter Jones' lab. And the animal model actually expressed DUX4 at very, very low level. It's so low, it doesn't even induce those downstream changes. However, these animals actually show muscle weakness and some pathology we see in muscular dystrophy. So we thought this probably is a good model to start with, because we want to see, even with this low expression of DUX4, whether we can do anything about it. So we first try to see if we can see that compound get into the muscle. So this is actually a fluorescent labeled compound, and then this A is a control, but you can see B and C actually they can get into the muscle tissue. Okay, and for the next several slides, I just want to show that after the first experiment, we actually did subcutaneous injections. So we delivered them several times within a period of time, and then check whether it can reduce DUX4 in vivo, so inside the muscle. So there are two different muscles collected here. One is the quadriceps and a thigh muscle, and the other one is gastroc, how do you call that? This part. And as you can see that the first bar is actually from the wild-type sibling. These animals do not carry the DUX4. They don't express DUX4. And then the second bar is actually from the animal that expressed DUX4. And the third one is actually after they received the treatment for 6 injections every other day. The most recent is a longer trial. We want to see if we can improve muscle function, make them stronger. So this basically just show you that in this trial, we can also knock down DUX4 to even greater extent. And this actually just to show that it actually improved the grip strength after 8-week treatment to the wild type level. So it's comparable to the animal that's not expressing DUX4. Okay, the last slide is an acknowledgement of all the collaborators participating in this project, and also most of the studies are funded by FSHD Society and Muscular Dystrophy Canada. Thank you.
Okay, I'll go quick because we want to leave time for questions. Okay, so I mentioned earlier that I've been focused on gene therapy for many years. I'm only going to tell you about one of the gene therapy strategies we have going in the interest of time, but I do want to mention and spend a minute or so just telling you about gene therapy. It is not it's not science fiction anymore. We actually have the first approved gene therapy on the market now. FDA approved it. It's actually a treatment for blindness. I'm going to show you that because I think it's exciting and it helps inform how we're thinking about treating FSHD with gene therapy. So a couple of the advantages of it are that, well, the major advantage of using gene therapy to treat any disease is that in general, it is a one-time treatment. So we're delivering genetic material that should, if it works correctly, correct the disease. And the way we're doing that is to use a modified virus. So viruses have had millions or hundreds of millions of years to evolve and figure out ways to enter tissues And so they know how to, there are certain viruses that know how to safely get into muscle, and we could use those to deliver genes that we want to use for therapy. Okay, so can you show the first video, please? 1A. So I mentioned that the first approved gene therapy is on the market now. It's a product called Luxturna. This is a treatment for blindness. And I'm showing this for two reasons. One, again, to demonstrate that gene therapy is a reality now. And the second, because we're using a lot of the similar tools that they're using here for blindness. So what we have here is a little boy who's blind. He's been treated with the gene therapy in one eye. And what they've asked him to do is they've covered the treated eye, and they've asked him to walk through a maze that has black arrows on the floor. And so you can see that he can't navigate. And if you heard this, you could hear the sound, he would say, "This is hard, I can't see anything." You can sort of see that type there. But basically this goes on for 3 minutes, and he never makes it through the maze. Can you shut this one off now, put on the other? This is a study that came out of the University of Pennsylvania and then was picked up by Spark Therapeutics out of Philadelphia. Okay, so in the second video now, this is the same exact kid just a few minutes later, and this time they've moved the patch from his untreated eye and moved it over to his treated eye and asked him to navigate the same maze. What's going on here? Is it not going? No, Can you delete the first video? Maybe it's just too big of a file. Here we go. Okay. I mean, to me, that's amazing. That's miraculous. Okay, so the point is that we've been thinking as a field about gene therapy for 40 years, and there have been a lot of hurdles, a lot of setbacks, and now we finally have the first treatment on the market. There are still a lot of hurdles that need to be cleared to actually make this feasible and make it widespread. I mean, they're treating the eye versus, an eye is a very small space versus an entire human being's musculature. So there are hurdles, but we are using the same types of viruses they're using here, and we're able to deliver these viruses to muscle. And so I'm just going to show you very quickly, because we're running out of time here, one of the things we're doing now in mice. So without spending too much time on mechanism, I'll just say that we can clone these small RNAs, similar to the antisense that Dr. Chen mentioned, but we can express these from a virus, and we can package those inside the virus. We sort of trick it into thinking that it's delivering its own genes, but instead of delivering its own genes, it's delivering our payload. We can deliver that to a muscle, either by intramuscular injection or actually through the bloodstream, and it will find the muscle. This particular virus likes to infect muscle. And this particular virus also, it's called adeno-associated virus, is not known to cause any diseases in humans. So it's a benign system. And when we deliver this, what we call a microRNA, this microRNA is designed to knock down the DUX4 gene, to reduce it. Okay, so we have a mouse model. I won't bore you with all the details again in the interest of time, but what I'm showing here are low power images of muscles from this mouse. We've purposely colored in regions that are damaged in blue. So that's a muscle section. The blue indicates muscle damage, and so those are those are two muscles from DUX4 mice, and then we have some controls that don't express DUX4, and they're pink, and that's what we expect to see, nice, healthy muscle. So when we packaged our microRNA system that knocks down DUX4 into this virus and delivered it to mice, I'll just focus your attention down to I guess it would be the bottom left. You can see this is the same mouse now. One muscle, so this is the calf muscle. One's been treated with our therapeutic vector. The other one has not been treated. And I think you can just look at the low power shot there, the two muscles. The blue indicates muscle damage. And so the untreated muscle has widespread muscle damage, and we can completely protect the mouse from muscle damage with our gene therapy vector. So I'll stop there, and that leaves us only a few minutes for questions. Unfortunately, we went long, and we have to be out of the room by 5 so they can set up for dinner. So we'll have to do questions now and then find the researchers, I guess, during dinner as well. So thanks for your attention. Sorry we went long. That's
Thank you.
my fault. I wasn't a bully.
So we have time for a couple of questions.
Hi all, I just wanted to say thank you for all your incredible work. It's really amazing to see, just even in the last 2 years. One question I did have was, do you think that targeting DUX4 or the mRNAs or even the protein, let's say you were to target the protein, you still have to use a lot of energy, time, and resources to transcribe the mRNA and then convert it to the protein and then get rid of it. Would you say versus doing it at the top, right at when you're in the DNA, I guess, would you feel the effects on your body if you were to take a drug that targeted the upstream versus the downstream effect? Does that make sense?
Anybody want to try that one?
I would say that it's difficult to predict that you would see a dramatic difference between inhibiting the production of the RNA versus once the RNA is made, breaking down the RNA, or even once the protein is made, impacting the protein. So you heard a lot of different approaches. You could block the production of the RNA, you could break down the RNA and stop it from making protein, or you could inhibit the protein. I would say that if any of those were extremely effective, they would look very similar in the outcome in a muscle.
This question is for Dr. Ati from Acceleron. So the ACE83 is specific to a muscle. Right now we're doing the leg and bicep. Assuming that's successful, we could move to the chest, back area. And what does that, that's the question. And what does that look like as far as approvals? Does that have to be retrialed, or is that something that's added on later? Does that delay the process? Could you speak to that, please?
Yeah, it's a great question. Generally, you get approval from regulatory agencies for what you study. And so our assumptions would be that if we could treat the TA and the biceps and see improvement in both, that we get approval for two muscles. Now we've gotten approval in an upper extremity and lower extremity muscle. Does that mean it would extend to other muscles? I might just answer that by way of Botox. Botox is being used for the opposite reason. It's used for spasticity to relax muscles, so we're sort of the anti-Botox. And they currently have a very broad approval on their label. It says the treatment of spasticity of the upper limb and lower limb. And in their studies, they used patients from various diseases, and they studied, I think, as many as 13 muscles. So now in their literature, they'll tell you how to inject each of these 13 muscles, some of them in the upper extremity, some in the lower extremity. So it's a question of how many studies you have to do to get approval for each of these. But what we're trying to show is that even muscles that have different architectures, some of them have, two portions of the muscle that come together. Some of them have a certain structure that's different than others. That regardless of the architecture of the muscle, we're seeing this enlargement. So hopefully it's a generalizable concept.
Thank you. This is incredibly exciting. But I want to just fast forward a few years. I think all of us in the room here would like to participate in trials and when, or in type trials, and when therapeutics become available, we want to maximize our chances of success personally. So suggestions on personal physical preparation, being ready for administration of treatments.
We have a couple physicians on the panel here. Any thoughts?
I mean, I could speak to the practicality of being in a trial. I mean, it means frequent study visits, and if you live a distance away, we heard that there'll be support for travel expenses, but still, if, for example, for our trial, if we want you to come every 3 weeks, that could be an issue. Some treatments take longer than others. Our treatment only takes a few minutes to do, so maybe, you know, on your way to work or school or whatever you're doing, it's doable. But, you know, then the MRI exams require scheduling and some time off to do the MRI. So I think understanding well what the study is going to entail, how much time it'll be, if it's a 6-month trial or a 12-month trial, how many visits that'll be. You might need to have, you know, your support people and helping you to get to and from the clinic. Otherwise, the inclusion criteria for the study will determine if you have the right characteristics, and it's something that we're going to have to think about as we talk about clinical trial preparedness. And is this for FSHD1 and FSHD2? Is it for mild to moderate patients or severe patients? And are there age cutoffs? So you'll see if you qualify.
Quick question, Celeron question again. Sorry, it's going to stand up. But question is in reference to once this initial clinical trial, I think it's 15 months, is over, what would you look at for approval from the FDA? How many more years would that take?
So that's a really good question. We've heard about orphan drug designation that helps you get the attention of different groups at the FDA that are interested in rare diseases. We also have what's called fast track designation. That means that we can have some more interactions with the agency to help us design our trials so that they know what to expect at the end of the trials. When we go to our so-called end of phase 2 meeting with the FDA, we're really talking about what is needed for approval, what are those outcome measures that you would consider approvable. The other thing I'll mention to you is that we have many of these so-called approvable endpoints already in a phase 2 trial. So there are times where you get very robust data and you can go in with these endpoints or surrogate endpoints. And we've seen situations where there's what's called accelerated or I guess in the US it's called accelerated, but it's kind of a conditional approval. So you get approval, but you have to do more studies afterwards. And so that's been the case for the Duchenne and the SMA drugs. So there's always the possibility of getting the drug to market earlier, but they want to see that you've shown some really good outcomes, and you take it from there. Otherwise, it would mean a traditional phase 3 trial, so maybe another 1 to 2 years of study.
One last question here.
This is for Monica. Congratulations on achieving the orphan drug designation. That's tremendous. And you identified some kind of target for 2019, but it wasn't clear to me what that was, what you're initiating by 2019. Could you provide a little more insight into that, please?
Sure. So our plan currently is to have a meeting early 2019 with the FDA. One thing I failed to mention about our drug, which I think is really important, is that we're actually repurposing a drug that's been in phase 1-2 clinical trials. It's been dosed much higher in that particular indication, so we think that our drug is going to be fairly safe at the doses that we plan to give to patients. So what we're planning on doing is spending most of 2019 putting together our application with the FDA with hopes that we will treat the first patients by the end of 2019.